So coming to the gram staining. This is the first differential staining used for group separation. It was first developed by Danish physician Hans Christian Gram in 1884. And this staining method was developed in Berlin. It's the most widely employed staining method in bacteriology. It is a type of differential staining procedure that divides the bacteria into four groups: gram positive, gram negative, gram variable, and gram non-reactive. So gram positive and gram negative, we all must have heard. And the other two categories are gram variable and gram non-reactive. So the reagents that are used for gram staining includes primary stain, that is a crystal violet, modern, that's a grams iodine, decolorizing agent, that is 95% ethanol or acetone, and a counter stain is used, that is safranin. So the gram staining procedure. In the step 1, we have a bacterial smear on the slide. We dry it and then it is heat fixed on the flame. Step 2, the smear is stained with primary stain, that is crystal violet, and it is allowed to react for 1 minute. Then step 3, stain is washed off with distilled water. After that, grams iodine is applied on the smear and allowed to stand for 30 seconds. It is used as a mordant. Grams iodine is used as a mordant that increases the interaction between the cell wall and the stain. So this is the function of the mordant which we studied earlier. So that is why the gram stain is used. Then the slide is decolorized with 95% ethanol until all the color is removed from the slide. After that, in step 6, counter-stain safranin was added and allowed to stay for 1 minute. In step 7, we wash off the bacterial smear of the slide with water, and then the slide is blotted dry and examined under microscope. So observation under microscope. Gram-positive bacteria will retain the CV, that is crystal violet, and appear purple in color, somewhat like this. Gram-negative bacteria will retain safranin and appear pink in color, like shown in this picture. Gram-variable bacteria will retain the stain unevenly and appear as a mixture of color, like in this picture, like shown in this picture, purple and pink. So, two types of color we can see in this gram-variable. Gram non-reactive, they retain the color very poorly, or sometimes they do not even retain color. So they appear like transparent in color. So there is a theory behind the gram non-reactivity, that is, the cultures that are too old, that is more than 48 hours of culture, if we stain it, then they will give this gram non-reactivity. So therefore it is recommended that we use 12 hours culture to 24 hours cultures maximum for this staining procedure. So what is the mechanism behind this gram staining? So the mechanism depends on the structure and composition of the bacterial cell. So there are two types of theories present. First is lipid composition and the second one is peptidoglycan composition. So first, coming to the lipid composition. In gram-positive bacteria, what does it have? It have a very lower lipid content than gram-negative bacteria. So that's why it dehydrates when treated with alcohol. And after dehydration, what happens? Pore sizes decreases, resulting in the reduced permeability, because of which the CVI complex, that is crystal violet and iodine complex, cannot be extracted out from the cell wall. So therefore it appears purple violet in color. Whereas in gram-negative bacteria, they have a higher lipid content because of the presence of outer membrane. In the alcohol treatment, what it does, it extracts the lipids. So the pore sizes increases, permeability is therefore enhanced, and the CVI complex is completely extracted out. Therefore, what they do, they retain the safranin and appear pink in color. So this is the first theory that we know. So the second theory is based on the peptidoglycan composition. So in gram-positive bacteria, they have a very thick peptidoglycan. This peptidoglycan is extensively cross-linked, and because of this cross-linked, pore sizes are very small, resulting in less permeability again. Therefore, the CVI complex is trapped and they appear purple violet in color. Whereas gram-negative bacteria, they have a very thin peptidoglycan as compared to the gram-positive. Therefore, they are less extensively cross-linked. Pore size remains sufficiently large. Permeability is high, and they cannot retain CVI complex. It gets washed out. Therefore, they retain safranin and appear pink in color. So, this will become more clear when we see the structure. So, coming to the bacterial cell wall composition. Here we can see gram-positive bacterial cell wall, gram-negative bacterial cell wall. So, here we can see that gram-positive bacterial cell wall has a thick peptidoglycan followed by periplasmic space underneath, then cytoplasmic membrane or plasma membrane. So this is the structure. We can see it's extensively cross-linked peptidoglycan. Therefore, the crystal violet is retained strongly. Whereas in gram-negative bacteria, there is an outer membrane present. Underneath, there is a peptidoglycan layer which is very thin as compared to the gram-positive bacteria. Underneath there is a periplasmic space followed by the plasma membrane. So here in the structure we can see the peptidoglycan is less extensively cross-linked, which upon dehydration, what it does, it extracts out the CVI complex. Therefore, it's retain the safranin color and appear pink in color. So this is the mechanism of the gram staining.